How do prokaryotes respond to sudden environmental changes? They induce synthesis of appropriate proteins, because transcription and translation are closely coupled. Process takes only minutes. Which respond quicker, prokaryotes or eukaryotes? Prokaryotes, because their transcription is closely coupled to translation. What is constitutive expression? Some proteins constantly needed for cell functioning, housekeeping genes, synthesized at all times. Examples of constitutive proteins: Tyrptophan repressor, TRPR, cap catabolite activating protein. Why are all genes not constitutive? It would be wasteful and there would be no adaptability for changing environment. What is an operon? Cluster of structural genes that encode for proteins that are involved in a related function, along with regulatory sequences regulating transcription of these genes, i.e., enzymes of biosynthesis pathy. Operon, simplified. Genes plus regulatory sequence. What is a repressor? DNA-binding protein that blocks the binding of RNA polymerase to the promoter. How does the repressor work? Repressor protein binds to specific DNA sequence called the operator that is located within a promoter at its 3' end. When repressor binds, DNA cannot be transcribed because RNA polymerase cannot bind to promoter. What are the two mechanisms of regulating the repressor? 1. Induction. 2. Repression. How does induction work? An inducer inactivates the repressor so it won't bind. DNA gene is expressed. How does repression work? A co-repressor activates the repressor so it binds. DNA gene is expressed. More on induction. Repressor is active until a small inducer, usually a nutrient or metabolite, binds to repressor and inactivates it. More on repression. Repressor is inactive until a small co-repressor, nutrient or metabolite, binds to repressor and activates it. Induction examples: lac operon. Lac repressor is always being expressed, inducer lactose is absent or at a very low level. Operon is often the situation. Lac induction with presence of inducer. Inducer lactose at high concentrations will bind to repressor, won't bind. DNA gene expressed. Operon is on. Tyrptophan Operon Control, repression. regulated by negative control, gene expression regulated by repression. Tryptophan repressor is inactive without its co-repressor RNA polymerase can bind to promoter and transcribe 5 genes for Trp synthesis. If level of tryptophan inside cell is high, repressor is active, binds to operator, blocks binding of RNA polymerase to promoter 5 enzymes not expressed. Tryptophan picture. When tryptophan levels are high, is the repressor active or inactive? What does this do to gene expression? High tryptophan equals active, repressor equals RNA polymerase can't bind, gene not expressed. What are some basic properties of the tryptophan repressor? DNA binding protein with helix-turn-helix motif, symmetrical molecular structure, acts in dimer or trimer form, binds to major groove in DNA. What is induction? The repressor is active and bound to DNA until a small molecule binds to the repressor and inactivates it. Operon is then on. Example: lac repressor. What is repression? Repressor is inactive, operon is on, until a co-repressor binds the repressor and activates it. Binds to DNA, RNA polymerase can't bind. Example: Trp repressor. What is a property of Trp that is common among other repressor proteins? Helix-turn-helix motif, binds to major groove in DNA in a dimer or tetramer manner. What is positive control? An activator can help mRNA polymerase to bind and transcribe gene. How is lac operon regulated by positive control? Upon starvation, low glucose, there is a high level of signal molecule cyclic adenosine monophosphate CAP. CAP forms a complex with CAP catabolite activator protein, and forms CAP complex. This binds to CAP site, facilitates mRNA polymerase to transcribe gene. More about lac positive control. What is made when lactose is present and glucose is absent? LacZ, beta-galactosidase, hydrolyzes lactose to GLOX and galactose. LacY, polymerase, facilitates movement of lactose into cell. LacA, unknown function, not important. Describe what happens when glucose is present but lactose is absent or very low in concentration. Lac operon repressed. Operator site is bound by helix-turn-helix motif. Repressors are active. Repressor blocks RNA polymerase. Negative control. Example: describe the situation when only lactose is available. Lac operon maximally induced. Lactose is converted to L-lactose, which binds to repressor and inactivates it when glucose low camp is present, which binds cap to form camp-cap complex, binds to cap site. Camp-cap increases efficiency of RNA polymerase transcription. Describe what happens when both lactose and glucose are available. Adenylyl is deactivated in presence of glucose, thus no camp-cap complex cap binding site empty. While the operator site may not be bound by repressor, still can transcribe without cap site. When both glucose and lactose are absent or at a very low concentration, what is the lac operon status? Off slide 13 of lecture 24. Is lac operon controlled by positive or negative regulation? Both positive camp, negative repressor. What happens to regulatory proteins when their ligands bind? Both change conformation, camp for capalar, lactose for lac repressor. What conditions must be met for lac operon to be maximally on? One, repressor must be removed. 2, cap activator should bind at the promoter. What two mechanisms regulate the Trp operon? 1. Repressor, operator indication, repression reduces transcription 70-fold. 2. Attenuation, termination of initiated transcripts. What is attenuation? Termination of initiated transcripts. What is the mechanism of attenuation? Process of translating short, incomplete polypeptide. When cells starved for Trp, genes for Trp are expressed maximally. Under less severe Trp starvation, Trp genes are expressed at lower levels. Attenuation can regulate transcription level by a factor of 8 to 10, combined with repression mechanism 560 to 700-fold. What is the leader region and where is it? Occurs between the operator and E sequences of the Tryptophan Operon. Attenuator. A specific regulatory sequence that, when transcribed into RNA, forms a hairpin structure that stops transcription when certain conditions are not met. Regions of Trp that can determine whether there is transcription or it is terminated. Pairing of mRNA regions 1 and 2 causes RNA polymerase to pause. Pairing of mRNA regions 2 and 3 cannot stop transcription. Pairing of mRNA regions 3 and 4 terminates transcriptions. What does pairing of mRNA regions 1 and 2 cause? Pausing of RNA polymerase. What does pairing of mRNA regions 2 and 3 cause? This makes it so transcription cannot be stopped. What does pairing of mRNA regions 3 and 4 do? Terminates transcription. Position of ribosome plays an important role in attenuation. When Trp is scarce or in short supply, Trp tRNAs are unavailable, ribosome stalls at Trp codons and covers attenuator region 1b. Region 1 cannot pair with region 2, instead region 2 pairs with region 3 when it is synthesized. DC, region 3, now paired with region 3, is unable to pair with region 4. When it is synthesized, RNA polymerase continues transcribing region 4 and beyond, synthesizing a complete Trp mRNA. What happens to region 1 when Trp is scarce? Trp tRNAs are unavailable and the ribosome stalls at Trp codons and covers attenuator region 1. Region 1 can't bind region 2, region 2 binds region 3, region 3 can't bind region 4. RNA polymerase keeps transcribing complete Trp mRNA. What happens when Trp is abundant and not required? Abundant charged Trp tRNA available, ribosome moves quickly and doesn't stall at Trp codons. Continues translating leader sequence ending at region 2. Region 2 then can't pair with region 3. Region 3 pairs with region 4 instead. Pairing of region 3 and 4 is the attenuator and acts as a termination signal. Transcription then ends before the Trp synthesizing genes are reached. When Trp is not abundant, what regions tend to pair and what is the consequence? Region 2 and 3, because stalls at region 2, because regions 3 and 4 cannot bind the termination signal, the entire gene is transcribed. When Trp is abundant and not needed, what regions bind and what is the consequence? One. When T